What if I told you the family tree for the most common life forms on Earth has been completely wrong for decades? Today, we're cracking the case on the secret history of viruses. Okay, so let's get right into this scientific puzzle. RNA viruses they are absolutely everywhere. They cause everything from the common cold to devastating global pandemics. And yet, for all their dominance, their deep family history, how they're all related, has been a total black hole in our understanding of life. For the longest time, it was kind of like this. Imagine finding three huge, distinct groups of people who all share the same last name, but there are no family records to show how they're connected. That was the exact situation for virologists looking at RNA viruses. They had them sorted into three main buckets, these so-called Baltimore classes. Now, these classes just describe how a virus makes copies of itself. Positive sense is the most straightforward. Its genome acts directly as a message. Double-stranded is a little more complex. And negative sense, well, that one needs an extra step. But the big question was always just hanging there. Did these three groups evolve completely on their own, or was there some hidden link that we just couldn't see?
Well, it turns out for years, investigators had only one clue. Just a single genetic fingerprint that even suggested a connection between this vast, diverse world of viruses.
And here's that clue. A single shared gene that connects thousands of wildly different RNA viruses, from the ones that infect plants to the ones that infect you and me. It's the closest thing we have to a universal genetic signature for them all. That master key is the gene for an enzyme. It's a bit of a mouthful, called RNA-dependent RNA polymerase, or let's just call it RdRp for short. This thing is the engine that lets an RNA virus make copies of itself. It's absolutely fundamental to what makes an RNA virus an RNA virus. But here was the problem. With only a small number of known viruses to look at, the evolutionary signal from this one gene was just too faint. It was like trying to solve a 10,000-piece jigsaw puzzle with only a handful of pieces. Yeah, you could see a few connections, but the big picture? Impossible to make out. That is, until a technological revolution basically dumped all the missing pieces right onto the table, all at once. And just like that, what was a tiny trickle of data became an absolute flood of new evidence. This huge breakthrough is called metaviromics. Think of it like a high-tech genetic dragnet. Scientists can now take a sample of, say, seawater or soil, or even an insect, and sequence every single virus hiding inside it. And this suddenly revealed an immense, hidden diversity of the viral world that we never, ever knew existed. So how much data are we talking about? The study we're looking at analyzed over 4,600 unique RdRp sequences. To put that into perspective, it's like going from trying to read a single torn page of a book to having an entire library pop up at your fingertips. For the first time, scientists had enough information to see the complete story. So, with all these new pieces on the table, scientists could finally reconstruct the family tree and crack the case of the RNA virome. This was the moment of truth. With this mountain of new genetic evidence, what did the complete, reconstructed family tree of RNA viruses actually look like? It revealed this, the new, comprehensive family tree of RNA viruses. Now I know, at first glance, it might look like a confusing mess of lines. But trust me, if you look closely, this image tells an incredible story that completely rewrites our understanding of how viruses evolve. So the new tree splits all RNA viruses into these 5 main branches. But the real shocker is that, as you can see, some of those branches are a mix of different Baltimore classes. That was the first major clue that the old, segregated model was just plain wrong. This new structure tells a coherent story, rewriting the entire origin of RNA viruses from the ground up. The tree's structure revealed something incredible. As this quote from the source paper puts it, the different classes weren't separate families at all. Instead, they were nested inside one another, kind of like Russian dolls. Double-stranded viruses evolved from within the positive sense viruses, and then negative sense viruses evolved from within the double-stranded viruses. So the crucial point here is that there's a clear evolutionary path from simple to complex. It starts with the positive sense viruses. They're likely the ancestral form. From them, the more complex double-stranded viruses emerged. And finally, from a branch of those, the negative sense viruses evolved. One group literally gave rise to the next.
And this diagram just beautifully illustrates the entire journey. You can see the simple ancestral forms on one side, and then this explosion of diversity as viruses evolved new strategies and moved into new hosts, everything from fungi all the way to us humans. So how did they pull this off? Well, viruses are the ultimate genetic thieves and traders. They are constantly capturing new genes from their host, shuffling and swapping functional parts with other viruses, and making these huge evolutionary leaps by jumping between completely unrelated hosts. And all this constant swapping, shuffling, and jumping is exactly why the RNA virome is a truly global network. It explains why we can find a virus in an insect that is surprisingly similar to one found in a plant. Viruses don't respect the boundaries between kingdoms of life. They actively connect them. So, we've solved the mystery of the viral family tree, but it leaves us with an even bigger, more profound question. If viruses are a planetary engine for moving genes around, blurring the lines between hosts, well, it forces us to reconsider the very nature of life itself and where one species truly ends and another begins.